Strums layered with awesome pads and a gentle tuck up beat and include the tones and subtle rising
That maintains perfect silence.
Curiosity, clarity in classroom. Awesome. Bright acoustic guitar strums layered with soft synth pads and a gentle tuck of beat. It includes low tones and subtle rising tones to convey curiosity, clarity in classroom. Insightful. Lo-fi jazz, a soft piano, brush drums, groove at subtle digital chimes to mimic
classroom
engagement. Perfect as a backdrop for introducing bioinformatics topics. Lo-fi jazz with soft piano, brush drums, and a gentle upright bass groove at subtle digital chimes to mimic classroom engagement. Perfect as a backdrop for introducing bioinformatics topics.
So once again, good morning. I want to really apologize for starting behind schedule. From exactly what happened, I want to make an analogy of what happened. Some of you will see that we have labored so much just trying to connect one wire to another one. And one of the reasons why we labored too much was because we did not have the necessary tool. Where we are supposed to use plier, we are making use of pen. So that is the same way. If you don't have the required tools in bioinformatics, that is the same way we are going to labor for a very long time. And that's the reason why what we want to do today, we want to consider different types of databases so that you will be able to know, depending on the data that is available for you. The data could be DNA, it could be protein, and it could also be some metabolic pathway. And that is what we are going to talk about today. And from what we have, it is introduction to biological databases. don't forget that bioinformatics, once again, is the analysis, retrieval, and utilization of biological data. And I told you that it's only dealing with informational molecules. Informational molecules from DNA to RNA, then to protein. So, what do we mean by biological database? It is a structured digital resources for storing biological data. So if anybody asks you what a biological database is, a biological database is a structured digital resources for storing biological data. And the purpose is to organize data for search comparison, to organize okay, to organize data for search, comparison, and analysis. And the type of database we will be making use will be the one that we can use for molecular information, the one we can use to determine their function, the one that can actually identify, that is in terms of taxonomic information, then the one we can make use of anytime we want to look at the image and we want to know at the specimen. Now there are two types of databases. The first one is called the primary database. The primary database, that is the level. The primary database are those ones that will contain DNA for you, because information, protein, will actually start from genbank. So that is why we have two types of database. We have primary as well as secondary database. The secondary database are the ones that are already curated. Curated means that they are specifically from the primary database. So you can see that without going to primary school, it is not too possible to get to the secondary school. So example of primary database is the genbank, and the example of secondary database, which is also called curated database, is UniProt. Now, what are the major biological databases? As a bioinformatician, as a molecular biologist, as a microbiologist, as well as even environmental biologist. Number one, the major databases are the genbank, which has to do with storing information that contains nucleotide sequences. Storing information that contains nucleotide sequences. PDB, that is protein database, which is where you can actually get three-dimensional structure, molecular structure of protein. And also UniProt, and that is where you can get protein sequences and you'll be able to determine their functions as well. Then you are looking at different pathways, metabolic pathways like Krebs cycle, glycolysis, synthesis of nucleic acid, lipid, and so on. You make use of KEGG. So, and other databases that you can also talk of are the DDBJ, EMA, REF sequence, as well as Metabolic. So all these are part of what is going to constitute to major biological databases. And from what you see, you will see that genbank contains information at the alphabet level. It contains information about alphabet level. Then the PDB has to do with the structure of protein, the structure of protein, and that is what you have here. And if you look at the structure of protein, it is either alpha plated, that is alpha sheet, or beta sheet. So that one is very, very important. And if you go to UniProt, UniProt is going to give you information that's going to tell you about where that protein is being located. Or the protein might be located inside the cell, that is cytoplasmic. It could be located outside, that is it could be membrane bounded, or it could also be located intra, that is in between the outer membrane as well as inner membrane. So all these are part of what you should know when it comes to determination of major databases. Now, what are the offline tools and access method that you can see? That is, what are those things that you can still do even if you do not have internet, even if you don't have any other thing apart from what you have? So number one, you can make use of genbank, PDB, that is protein database, as well as UniProt. So you can do that when there is internet. You download all these, then later on you can also perform local blasts. So local blast, whereby you do search offline against different types of genome. Then you make use of spreadsheets in order to visualize and filter sequence information. Then you make use of a biopython, which is actually used to automate faster, as well as a genbank passing. Then you can also make use of a PyMore. All these you can download them even before you get to making use of all these tools. So you need to know this. Now, what are the do it yourself offline data retriever? That is, what can you do on your own without even necessarily being on internet? The first one is on genbank. You can download. If you download, you open, and you can also do the analysis. Then the other one you can do is you go to UniProt, you also download, you can open, then you analyze. Then the analysis that you are going to make is what you can use for metabolic pathway. So, and I've mentioned that to you. And the practical exercise that you can do, number one, at this level you should be able to know how to run blasts, making use of microbial sequences, Then apart from that, you analyze the gene bank using CDS format, and you should be able to translate what's the problem over there. Okay, then you should be able to make use of a PyMore. Some of you that are working using your Android phone or smartphone, you might not be able to, not all phones that can actually download PyMore. So that means you might not be able to work it unless you move close to somebody that has it. Then you do extraction of your protein, and you can even load it into from that UniProt offline into the file. Then you can do alignment of sequences with cluster omega. And we have different types of case studies that you can do. That is, whether you are in environmental or you are in microbiology. If you are in environmental, you make use of pollutant degradation. That is, what you do is that microorganism of your interest, you just go there and go and retrieve the sequences in form of the genes. By the time you retrieve the sequences, you compare the GC content. By now you should know what GC content means in, okay, that GC content. Then we'll be able to do antibiotic resistance using KEGG plus RGI. KEGG means comprehensive antibiotic resistance databases. That is the database that you can get information about some microorganisms that will develop resistance in the long run. Then you make use of metagenomics by identifying unknown sequences, and you'll be able to make use of your basic local alignment sequence tool in order to do that. Then you make use of pathway mapping, which can be used for nitrogen cycle using some enzyme in KEGG. So then you should be able to understand file formats. Whenever you see a faster format, you should be able to understand that. The faster format, I've told you that it will always start with greater than sign. Then there's going to be an header to that. So and the gene bank, how you can understand whether an information is in faster format or is in gene bank. The gene bank will have a full annotation, which is going to include the CDS plus the note. You have an example like that in the screen for you. Then PDB format will always have a 3D coordinates using Pymon, that is, it is a rotational, where you can know at the angle at which to rotate. Then whenever you see IDS, that is the identity, you are trying to discover the identity, it is going to be your gene bank, comma, your UniProt. So if it is gene bank, it is going to start with NM, or it's going to be capital letter that is going to be 2. Then the number can continue. If it is a UniProt, it is going to start with P. You need to know that. And if it is a PDB, it is going to start with a 4, and so on and so forth like that. Then as a general question now, you should be able to define primary and secondary databases. If somebody asks you to mention what are the primary databases and what are the secondary databases, then GenBank, if you are working with GenBank, you should be able to download and annotate lactZ gene, lactZ, that is lactogen, or lactose Z gene, that is the gene that is responsible for metabolizing lactose. So you will see that. Then we are looking for match format. By now, if you see a faster format, you should be able to know. If you see a gene bank format, you should be able to know, or gene bank number, you should be able to know. PDB format, you should be able to know. Then if you want to also do blasts, by now you should know the workflow. Where are you going to start? If you want to do blasting, don't forget that you are going to start with NCBI. When you start with NCBI, you type in the gene you are actually looking for. If it is a 16S ribosomal RNA, you type it there. And if you are looking for this, then you make sure that under where you have all databases, you put it in. If you are working with protein or amino acid, that is what you are looking for. You make sure the all databases, the drop down menu is in that. So put it there. If you are working with DNA, put it on nucleotide. If you have RNA, if RNA is what you are looking for, you put it at still nucleotide. And if it is protein, put it at protein. Then you do the search. After doing the search, you retrieve the information, that is the sequences. You retrieve it in faster format. And when you retrieve it in faster format, then you now continue to do a kind of further work on it, and that's going to lead you to do blast on that. Then you will be able to talk about the workflow, as you know. Then when there is no internet, what are those things that you can do offline? By now you should be able to know that. Now the DIY, which is going to be the second aspect of this, that all of you will do, and that's what you are going to do today. That's going to be your CA. It's going to be do it yourself manual. Do it yourself manual. So you are going to take a step by step offline guide for you. So you are going